Without a doubt, one of the most serious and deadly pathological threats to the world population is the human immunodeficiency virus, more commonly known as HIV. Over 34 million people throughout the world are currently infected with this virus. The HIV-1 virus, the most common viral strain, is a retrovirus that causes immunodeficiency by targeting a select group of cells that express a glycoprotein known as CD4. CD4 is expressed on the membranes of dendritic cells, macrophages, and, most importantly, T helper cells. T helper cells serve an important function within the immune system because they are responsible for identifying a variety of foreign antigens and prompting the appropriate immune system response. The other cells also serve vital roles within the immune system. However, HIV-1 is able to hijack this line of defense by binding to the CD4 receptor because of a glycoprotein on the HIV-1 envelope known as GP120. GP120 is a surface protein and remains bounded to the membrane through a transmembrane protein known as GP41. Three GP120 proteins and three GP41 proteins organize themselves into a trimer on the membrane in order to form a spike-like protrusion on the surface of the envelope. The following image shows the organization of three GP120s arranged in their trimer structure. This viral spike binds to the CD4 receptor and initiates a series of conformational changes that cause the virus to fuse with the membrane and enter into the cell, leading to T-cell death. The progression of HIV to AIDS occurs when the cell count of these T helper cells becomes significantly low, allowing for opportunistic infections to cause death. Therefore, any antibody that can block this infection cycle will neutralize and prevent HIV-1 viral progression. The main problem is that the HIV-1 envelope has a high mutation rate, making it a difficult target for identification by the immune system. Therefore, only a broadly neutralizing antibody that targets a conserved region of the virus can effectively combat most HIV-1 isolates. The problem is that most antibodies naturally produced against HIV-1 contain little neutralizing abilities. Only a small percent of patients are able to actually develop broadly neutralizing antibodies. Therefore, in a study published by Zhuliang Wu, scientists designed a recombinant protein known as RSC3 that preserved the CD4 binding site on GP120 for easy identification by an antibody. Sera collected from infected patients were then tested for its binding affinity to RSC3, and one donor, patient 45, had sera containing broadly neutralizing antibodies. After sorting through the antigen-specific B cells within this sera and using PCR to amplify antibody expression, an antibody known as VRC01 was found, which could bind to the CD4 binding site. The antibody's binding efficiency was then tested using ELISA assays against 1,990 HIV-1 viral strains, and it was able to neutralize almost 91% of the isolates. Like most antibodies, VRC01 is a heterodimer composed of two polypeptides, each with a heavy and light chain. The binding site on VRC01 is similar in structure to the CD4 receptor, and by binding to the GP120 glycoprotein, it prevents the virus from infecting other T helper cells. The binding occurs through similar ligand-protein interaction forces as seen in the interaction between GP120 and CD4. The following image shows the VRC01 antibody in complex with a monomeric trimmed GP120 protein. The image shows the inner and outer domain of the GP120 protein along with VRC01's heavy and light chain. The main points of contact on the GP120 protein are the structures labeled as the CD4 binding loop, the loop D region, and the loop V5 region. The corresponding points of contact on the antibody are labeled as the CDR regions. The next image shows the similarities between the GP120 VRC01 interaction and the GP120 CD4 interaction. Note the similarities in secondary structure, and the close-up images even show a similar hydrogen bonding structure at 2 amino acid residues in both complexes. Clinical trials in non-human primates have already demonstrated this antibody's ability to provide immune resistance to SIV, the simian equivalent of HIV. By manufacturing this antibody and administering it to patients through immunotherapy, or finding a way to produce this antibody naturally through a modified vaccination plan, the antibody could confer immune resistance to HIV. Thus, further research into the VRC01 antibody is of the utmost significance, since it could provide a way to curb the worldwide viral epidemic of the HIV virus.